So is now the mechanism by which this antigen presentation process occurs. So the questions I want to focus on first is, what is this? Oh, with the spare one. Oh, here it is. So you have to thank Zoe, because Zoe now has a spare. What is the structural basis by which the Mhc molecules present peptides to T-cell receptors? Namely, basically, what is the structure of the MHC? How is class 1 MHC different from class 2 MHC? And how does that structural difference impact on the function of these Mhc molecules? How are endogenous peptides targeted to class 1 Mhc molecules and exogenous to class 2? There must be some kind of sorting process that goes on inside the cell. What exactly is that sorting process? Also, you appreciate that putting a foreign peptide in a class 1 Mhc molecule is a death sentence. And therefore, it's critical to put the right peptide in the right Mhc molecule. Otherwise, you will kill an innocent cell. And how does the T-cell receptor see the peptide in the Mhc molecule? Is it seeing just peptide? Is it seeing some MHC? What parts of the peptides exactly is it seeing? And finally, what is the structural basis how CD4 T cells interact with Mhc class 2 and why CD8 T cells interact with Mhc class 1? So again, this is just, you've seen this picture before, again, to show it again, but just to review the kind of core interaction. And we'll now start breaking down and focusing on the structure of the Mhc molecules. Mhc class 1 interfaces with T-cell receptor and CD8. Now, just looking at this cartoon, you could immediately start seeing things that we'll pick up on as the lecture unfolds. The first is, if you look at class 1 MHC cartoon structure, you have basically this long alpha chain having an alpha 1 domain, an alpha 2 domain, and an alpha 3 domain, and this is anchored into the target cell. This is associated with a protein called beta 2 microglobulin. And look, if you look at it, beta 2 microglobulin is not interacting with the cell membrane at all. It's basically interfacing with the class 1 MHC molecule. Now, the class 1 Mhc molecules I'll discuss in a few minutes, the alpha chain, there are many, many, many different sequences for this alpha chain, but the beta 2 microglobulin is exactly the same. And in fact, my beta 2 microglobulin protein is exactly the same as pretty much everyone in the room. But my Mhc class 1 is going to be different from that of the overwhelming majority of people in the room. So this is, one thing we'll focus on. In addition, you'll see here how the peptide is floating inside this little pocket. When it's by floating, it means there's no covalent linkages. This peptide is not covalently linked to the Mhc molecule at all. It's only being held in place by hydrogen bond interactions or other ion-ion interactions. It's kind of like floating in this magnetic field, but it's not physically being linked to it at all. So it could sometimes even be popped out of this cleft, and that's an important point that needs to be appreciated. If you look at the T-cell receptor, this is the hypervariable region, and what is it seeing? Well, first of all, you see both the alpha chain and the beta chain are contributing to recognition, and you see that each one of these is picking up a piece of the peptide, and each one of these is also recognizing the Mhc molecule. But what this is also underlining is, as I mentioned before, the same alpha chain with a different beta chain can recognize a different peptide, because you can see it's picking up different pieces of the peptide. Is that kind of clear? And if we now go to the class 2 Mhc molecule, you see a different structure. Here you have an alpha chain and a beta chain, so it's a heterodimer. And again, when I was a student, professors would talk about homodimers, heterodimers, and I would nod my head like I knew exactly what they were talking about. I was clueless. I didn't know what a heterodimer was. I didn't know what a homodimer was. And again, it's very simple, though. A homodimer means that both chains are exactly the same, like identical twins. That's a homodimer. A heterodimer is like fraternal twins, that each chain has a completely different structure, but the two chains combine together to interact. So this is a classic heterodimer. Two chains, they're different in structure, but they interact, so it's a heterodimeric chain. So you'll hear that all the time, heterodimers and homodimers, but that's what it means. But here you now see that the peptide is being held in place a little bit with the alpha and a little bit with the beta, so both of them are contributing to it. And again, the T-cell receptor is seeing some peptide and some Mhc molecule. But as we'll develop, this is being held in place by a single chain, and the structure of the cleft is going to be much more rigid, whereas this is being held in place by two chains. So think about, like, if you grab onto something with one hand, it's going to be very, very tight, but if you grab onto something with two hands, it's a lot looser, and this has tremendous impact in terms of the function of the Class 1 MHC peptide interaction and the Class 2. Again, I'll discuss in greater detail. Again, this is just kind of like a little foreshadowing. Now, there are some people who basically think everything can be understood through genetics. And there's obviously a lot of truth to that. So let's look at what the genetic basis for the MHC molecules are. So there are at least 3 MHC molecules for Class 2 and 3 for Class 1. Class 2 is a lot more complicated. But if we just think in terms of DR, DQ, and DP, because of the fact that each one has an alpha chain and a beta chain, so DPB is the beta chain, DPA is the alpha chain, what this is telling you is the number of alleles per locus. So for the DP beta chain, there are 120 different genes that can encode for that, for the alpha 23, et cetera, et cetera. If you go now to Class 1 MHC, you're obviously only seeing one bar because you only have one chain. The beta 2 is exactly the same for everything, but you have HLA A, B, and C, and each one of those has, again, tremendous number of potential genes that can encode for that locus. Well, what does that mean? There are 728 possible HLA-B genes. And again, many of you are studying like B57, B27, you know that has implications. But what this is telling us is in this room, the chances of two of us having exactly the same HLA-B is very, very low. It's probably minuscule. And similarly for A and C. So this is a high level of polymorphism that occurs within the HLA molecule. But again, each one of us has 2 chromosomes, which means that we potentially can express 2 different alleles encoding an A chain, a C chain, a B chain, each one of the alpha and beta chains, et cetera, et cetera. So that also contributes to our ability to express a wide range of HLA genes. Now, I mean, kind of like now, what's the difference between HLA A, B, and C? They're all Class 1 MHC molecules. The answer is I don't think we really know yet. I mean, we're getting some clues. I think HIV is really going to be very instructive because as work that Bruce's group and other groups have shown, B57 we know is highly protective for HIV. Why is that? HLA-A2 doesn't seem to be as protective. We don't really know. So clearly there's a lot more subtlety to Class 1 MHC A, B, and C that we know about that we're first learning. But anyway, the bottom line is that A, B, and C all encode proteins that have the capacity to present endogenous peptides, and all these different genes have the ability to encode exogenous peptides. There are clearly variations within, so the sequences of A are very different from B and very different from C, but all of these have exactly the same function. And again, you can think about this as, I don't know, strains of flowers or whatever, that A could be roses, for example, but within that rose family, you have 50 different roses. B can be, for example, a tulip family. They're all flowers, but within each one you have some functionals that are unique to them. And again, I think it can be a very exciting area determining differences between these structures. So what does this mean now from a genetic point of view is that if you're heterozygous for every HLA locus, which means that you could express 2 different genes for each HLA locus, and clearly this is the old days when women used to wear skirts, but clearly, clearly by having 2 pairs of pants on, it'd be a little confusing. But and they may have set genetics back a long time. But parents basically combine their genes. You have basically 4 different possible recombination events among the kids. But what you're seeing is that every gene is expressed on the surface at the same time. And this is using the genetic concept of polygeny, where you could have a lot of several genes, but on each locus is exactly the same, or polymorphism, where each locus on the chromosome encodes a different allele, a different gene, so that there's differences. So this could be HLA A, this can be B, and this can be C, but what is happening is that your cell is expressing 6 different HLA molecules at exactly the same time. Similarly, the same thing is happening for class 2 Mhc molecules. You're expressing, if you're heterozygous for every locus, at least 6 class 2 Mhc molecules at exactly the same time. So your cells are, again, expressing at exactly the same time all of the HLA molecules that you have genes to encode. And that has implications when we start discussing anchor motifs. Okay? So that's a very important concept to nail down, that your cells at any given time are expressing all of the class 1 MHC genes they possibly can express. And if you're heterozygous, that's at least 6 different combinations. And if you're a antigen-presenting cell, you're also expressing all at least 6 different possible class 2 MHC molecules. So you have a lot of different antigen-presenting molecules being expressed on the surface at any given time. Okay, is that clear? Okay, so we'll start with class 1 MHC molecules. And the class 1 MHC molecules, they can present a diverse yet limited number of peptides that are 8 to 10 amino acids long. And, well, okay. So the point, though, is what is the structural basis that limits the peptides that the MHC molecule can present? So can an MHC molecule present any possible peptide? The answer is absolutely positively no. They only can present a limited number of peptide. But the question is, what is the rule that determines what peptide an MHC molecule can present? And that's what we're going to start talking about. Well, let's first go to the structure. So if we look at the structure of the MHC molecule, the first thing, as I started talking about before, it's a heterodimer with a membrane-spanning alpha chain bound to the cell membrane over here, and a beta-2 microglobulin that is basically linked directly to the alpha chain. The alpha chain is polymorphic. It means that different genes can encode different alpha-2 chains, whereas the beta-2 microglobulin is exactly the same for everybody. Now, where does the peptide sit? The peptide is going to sit, if you look at the alpha chain, it has 3 domains: the alpha-1 domain, the alpha-2 domain, and the alpha-3 domain. And the peptide is going to be sitting in the cleft formed by the alpha-1 and alpha-2 domain. And as I had mentioned before, the alpha-3 domain is where the CD8 molecule is going to bind to. The beta-2 microglobulin, it's unclear exactly what it does. The simplistic view is it kind of shores up the structure of the alpha chain, because in the absence of beta-2 microglobulin, it's a very, very floppy chain and really can't hold on to peptide well. If you knock out beta-2 microglobulin in mice, for example, they really express extremely low levels of class 1 MHC, and functionally they're CD8 functional deficient. So this clearly plays a critical role, but it's kind of maybe like a doorstop or something that you stick in order to stabilize it. Now, this is a view of looking down on the MHC molecule. So now think of yourself as the T cell receptor coming into the cell to dock into the molecule, and this is exactly what you're seeing. This is the MHC molecule. If you kind of think three-dimensionally, these alpha helix coils kind of look like almost like a shark's teeth. And inside of it is the peptide floating inside. And you're coming in now to dock onto this peptide. And what we'll start discussing is pieces of the peptide point downwards towards this pocket of the MHC molecule, and some of the residues in the peptide point upward towards the T cell receptor. Another point to appreciate, if you could see these blue like dotted lines here, this is demonstrating that the peptides are bound within the cleft of the MHC molecule. This is almost sealed so that the peptide has to be completely inside this cleft. You can't have any peptide sticking outside. Again, the MHC molecule may be a little bit anal. It has to make sure that the peptide exactly fits. If it doesn't fit, it can't deal with it and won't be able to bind it. And again, this has important implications because this means that the peptide is bound very, very tightly. Once that peptide is in there, it's not popping out. It doesn't have dangling ends that could get ripped away. It's really tightly fixed there. And again, if you'd want to ask teleologically, why do you think that is? Why should the MHC class 1 peptide, would you want to have it bound so tightly that it can't pop out? Any suggestions? Well, what would be a ramification of putting a wrong peptide into a class 1 Mhc molecule? What could go wrong? The cell can be killed. So you're an Mhc class 1 molecule, and you have a self-peptide in it, and then all of a sudden a digested foreign peptide floats by. You pop out your self-peptide, the foreign peptide gets popped in, now a CD8 cell comes by, and you're saying to the CD8 cell, whoa, don't kill me, that was an accident, that's not my peptide, I'm not infected. Is the CD8 cell going to say, oh, sorry, never mind, I buy that? No, it's going to get killed. So it's really important for the cell to be totally paranoid about making sure that only peptides coming from inside the cell are in its class 1 Mhc molecule. It doesn't want to be an innocent bystander and get killed. So that's why class 1 is really very important, to have it tightly bound and not be able to be exchangeable. Okay, so now if we now do sequence analysis and we ask the question among all those multiple genes for class 1 MHC, and what are the constant regions and where is the hypervariability in the different genes that are out there? And these red lines indicate areas of hypervariability. These are relatively constant. And when you now map it out to the structure of the class 1 MHC molecule, lo and behold, the location for the variability turns out to be exactly in the peptide groove. So in other region, the alpha-3, for example, there's almost no very minimal variability. And in fact, now if you focus on where in the A2 and alpha-1 and alpha-2 the variability is within the areas that actually come in contact with the peptides. So this is going to be teaching us something incredibly important about the ability of different MHC molecules to bind to different peptides, because this is where the variability occurs, and this is going to impact on the range of peptides that any given MHC molecule can bind to. And that is determined by anchor residues that are present in the peptides. So if you look, and this is always confusing the first time you see these kind of ball structure diagram, but think of these as basically showing residues from the amino acids, and focus on and this, to clarify this is the outside towards the T cell receptor, and this is the bottom of the MHC molecule pointing down towards the cell. And if you focus on these residues, the P2 and the P5 and P8, so these are pointing downward, and these are providing what are called anchor motifs. And what is happening is that these stay the same, but these all change. And what's actually binding in the MHC molecule is these three particular residues, because there are matching pockets, which I'll show you in a few minutes, that line up with these residues that allow it to sit inside the MHC molecule. And what you now see is that for this particular K of B MHC molecule, these are all peptides derived from different proteins and viruses that can bind to K of B MHC molecule. What they all have in common is in the P2, the P5, and P8, they have pretty much similarly charged peptides. In this case, leucine and isoleucine. In this case, I always forget what S and Y stand for. But this is lysine, lysine, threonine. I'm blanking on the amino acid code, but these are relatively conserved, and these have to be the same in order to bind to MHC K of B, but these all can be totally different. And therefore, what allows an MHC, and now if you have a different MHC molecule, it has a different anchor residue motif, in this case the P2, P5, and P9 location of the peptide. And again, this anchor motif is different from this anchor motif, and this means that this particular MHC molecule can bind one range of peptide, and this one can bind another one. If you're not an anchor residue, it could be completely different, and that's what gives the tremendous variability for what can bind to an MHC molecule. Is that clear? I mean, I'm sure it's a little fuzzy for some people, so I'm going to have more reiterate this again in a second. And as I mentioned before, the fact that the peptide is limited in terms of how long it is, that means it's bound very, very tightly. It can't stick out of the cleft. So the ends are like almost sealed down by hydrogen bonds to make sure it's really staying in there extremely tightly. Different MHC molecules have different structures, and therefore it allows different groups of peptides to be contained within this cleft. So now, looking at it at a very two-dimensional point of view, what you have are one group of residues in the peptide point down towards the MHC molecule. These are the contact residues. These are the anchor motifs that allow this peptide to bind to the MHC molecule. These have to be relatively conserved at these locations. However, the other residues in the peptide, it can be any amino acid you want, any of the, 22 amino acids they can be, which means, therefore, if you have Y, Y, L in these locations, you could be presenting hundreds and hundreds of different peptides, all of which have different sequence residues at these different times. And what is recognizing these particular residues? What molecule? The T cell receptor. And what that means is that any given MHC molecule can express thousands of different peptides that can be recognized by thousands of different T cell receptors. Is that clear? And this is just another way of showing it, basically showing that the T cell epitopes are pointing up and the anchor residues are pointing down, and you could actually see they have these clefts inside of the MHC molecules that they're bound to. Different MHC molecules have different kinds of clefts, and therefore they'll use different anchor motifs. So now to put it together physiologically, what that means, though, is that if I would just have one MHC molecule, that's all I would have, I would really be limited with how many peptides I could present, because I'm limited to whatever anchor motif that particular MHC molecule has, right? So how do we get around that? We get around that by having not one MHC molecule, not two, not three, not four, not five, but 6 possible MHC molecules with 6 different anchor motifs, which means that when among all 6 of those MHC molecules, if you're heterozygous, you could now present a very wide range of peptides, and therefore hopefully present almost any possible peptide to a T cell receptor. And now you understand why we have so many MHC molecules, why we have so many genes for MHC molecules, because I guarantee you there are some peptides I can't present, but you could present. And again, if you think in terms of humanity, if I could not present peptides from a virus, what do you think would happen to me when I get infected with that virus? I'm going to die. So what humanity has done is basically it's said, let's make hundreds of different MHC molecules, so if some new virus comes, maybe we'll wipe out 90% of the population because they can't present it, but we're guaranteed that somewhere in humanity is somebody that potentially could be able to present a peptide and be able to survive. So maybe the B57 HIV story may be relevant in terms of that being super able to present it. Again, continue now being worked on. But now I think you have an understanding of why we have so many MHC molecules and why all of them are always being presented at any given time on the cell. Okay, is that clear? Okay, so now if we shift gears to Mhc Class II molecules, and Mhc Class II have similarities, but also have differences from Class I MHC. So they also can present a diverse yet limited number of peptides, but in contrast to Mhc Class I, which are relatively limited in the size of the amino acid, 8 to 9 amino acids, Mhc Class II are very liberal. They say, 13, 15, 16, no problem, I'm more than happy to present it. It's not anal compulsive at all. Well, why is that? What is the structural difference between Class II and Class I Mhc molecules? And, well, first of all, as I alluded to before, it basically consists of 2 dimers, an alpha dimer and a beta dimer. Both of them are anchored in the membrane, but both of them probably have a little bit more wiggle room in terms of interaction. And also, it turns out, when you look at the peptide binding cleft, instead of these 2 kind of being sealed together, these are like kind of opened up. And the example people talk about is more like kind of do you have frankfurters here in South Africa? Hot dogs, right? So think of a hot dog bun. You know, the hot dog is always sticking out the ends. I mean, imagine if you had a hot dog bun, you had to get exactly right size hot dog, you know, and otherwise it wouldn't fit. You know, it wouldn't go over very well. So this is pretty much what the cleft looked like. It's a little bit different. And now if you look at the peptide's sitting inside of it, but it's wide open, and you can actually have peptides dangling out in the breeze. And again, as I'll show you in a second, this has important implications in terms of anchored motif binding. Okay, now, so the possibility exists, therefore, that class 2 MHC peptides can fall out and new peptides can pop in. Does that bother you? Why doesn't it bother you?
Because Mhc Class 2 works together with CD4.
Right.
And they're not necessarily going to kill the cell if they see.
Right. They're not killer cells. So the worst thing that can happen is, you know, you could help a different T cell receptor, but you yourself are not going to get damaged. And actually that may be advantageous for you, because let's say there's foreign peptides floating around, you haven't phagocytosed that particular bacteria, processed it, but now you want to just stick those peptides into your MHC to present it. That's fine too. You're maximizing your ability to present peptides, which is good for class 2 MHC. The more peptide presentation, the more help, the better. So in fact, that's why the MHC class 2 does not have to be anal compulsive about making sure the peptide that's in it has to come from inside, and therefore the rules of binding are a lot looser than they are for class 1 MHC molecules. And in fact now, this has important implications in terms of anchor motifs, because class 1 and class 2 MHC both use anchor motifs. So in this case, position 1 is hydrophobic, position 4 is negatively charged, and position 9 is hydrophobic. These are the anchor motifs. But look what's happening. What's happening is that all of these peptides can be presented by exactly the same MHC molecule. But what you do with the peptide is you move the peptide back and forth in the pocket to enable those anchor residues to bind. So if it's located at the end, at the I guess this would be the C-terminal end over here, so that's fine, and then it sticks out over here. Or if it's here, you stick it out over there. You have a lot of movement you could do back and forth. With the MHC molecule, since the structure is limited, you can't move it back and forth. So you have less capacity to present peptide. But also it explains why you don't have to be so concerned how big the peptide is, because there's a lot of room to pack. I mean, if you go on a trip and you have a giant suitcase, you just throw everything into it because you know there's plenty of room. So too, class 2 MHC molecule doesn't have to be as concerned how big the peptide is. Is that clear? Okay, so again, to kind of reiterate, class 1 MHC, CD8, alpha beta, class 2 MHC here, heterodimer of the alpha and beta chain, again being presented to CD4. So what basically now provides you with CD4 or CD8 specificity, as I had mentioned before, is the ability of the class 1 MHC molecule or the class 2 to interface with CD8 or CD4. So here what you see is CD4, again, binding to the beta-2 region, and here the CD8 is binding mostly to the alpha-3 region, and that's what's providing the specificity. And again, what's really interesting is you could take a T-cell receptor that recognizes a CD4 peptide, swap out the CD4 for the CD8, and change it into a CD8 cell, and now it'll only be able to recognize it if a CD1 molecule is presenting. So the specificity is not determined by the peptide, it's solely determined by CD4 versus CD8. Okay, is that clear? So now, kind of to summarize, and class 1 MHC molecule, in terms of its structure, alpha chain beta-2 myoglobulin, whereas class 2 is alpha chain beta chain, peptide size limited to 8 to 9 amino acids, whereas class 2 is much broader range of size it could be. The cleft of the MHC, the peptide must be within the cleft. Class 2, the ends of the peptide could dangle. Binding affinity, class 1 is tightly bound, because if you bind the wrong peptide, it's a death sentence, whereas class 2, it's loosely bound. And T-cell interaction, class 1 is CD8 and class 2 is CD4. Any questions? Okay, so now the next question is, how do peptides get into those clefts? And what are the functional ramifications of this process? And again, as we've said over and over again, presentation of a foreign peptide in class 1 is a death sentence. So it's really critical to make sure that you put the right peptide in the right MHC molecule. So how does this happen? So cells have two intracellular compartments. One, which is the vesicular, communicates with the extracellular fluid and is continuously basically bringing in fluid from the outside, processing it and sending it outside again, whereas the cytosol is not in communication with the outside environment. It's kind of basically a sealed environment. And therefore, what that's telling you is that any peptide that's coming from the cytosol is an endogenous peptide, whereas any peptide that's coming from the lysosomes or from the endosomes are exogenous peptides. They're coming from outside. So again, this is now going to be telling the cell whether it's been permanently infected, because once the infection's in the cytosol, it's permanent, or is it just bringing something from the outside and there's no need to worry about killing the cell. And this is, again, just a slide I showed you the previous lecture, showing, again, cytosol pathogens, therefore, are going to be presented in class 1, seen by CD8 cells, and they're killed, whereas intravesicular pathogens, which means that it's not a terminal infection, class 2, CD4 help to induce killing, or B cells will be presenting internalized antigen in order to recruit T cell help to stimulate B cell class switching. So what compartment it's located in determines what it sees. So peptides that are presented by class 1 MHC molecules are derived from intracellular proteins. And to basically take the process, you basically start by having intracellular antigen. It comes from inside of the cell. It gets processed, as I'll show you in a minute, in a protein called the proteasome. And the proteasome basically slices this protein into the appropriately sized peptides. These peptides are going to be loaded in class 1 MHC molecule. And the class 1 MHC molecule cannot leave the endoplasmic reticulum unless it has a peptide inside. Once a peptide is loaded, it can now get cycled, it'll leave, and it'll get expressed on the surface. But this peptide ultimately is coming from inside the cell. If you're not infected, all these peptides are self-peptides. Nothing will happen to the cell because there's no CD8s that recognize self-peptides, hopefully in the outside environment. Question?
Is the proteasome an intelligent cutter?
Okay, excellent question. So, proteasome. Anyone ever see an egg slicer?
No.
No? No. Okay, so when I was when my kids were growing up, you want to give your kids food that's as easy to make as possible, right? Because you want to just quickly throw food on the table. So what's better than a hard-boiled egg, right? Just throw it in whatever, 3 minutes to boil. So, but then you got to slice it. So these egg slices are really cool because you put the egg in it, and there's these wires here. They're exactly spaced at exactly the same distance, and then you teach the kids that the only way they get to use this is if they eat the egg. And they love doing this, so therefore they say, please, can you make me an egg? Okay, if you insist. So then now you take this thing, you push it down, and now voila, you have exactly the same size slices of the egg that you've generated. So addressing your question, the proteasome, intelligence is a relative concept, but they apparently have the capacity to really slice peptides in relatively equivalent sizes to give you that 8 to 9 amino acid difference size that you need to efficiently load class 1 MHC molecules.
But when that 8 to 9 doesn't sort of like things P3, 5, and 8, doesn't it have to be 3, 5, and 8 in those specific?
Yeah, so the proteasome has no idea what your anchor motifs are. So the possibility absolutely exists that it may be off. So if it had sliced it over 2 amino acids over, maybe the anchor residues would be in the right location. And again, clearly, there's some proteasome is basically probably wiggles the peptide up and the protein up and down a little bit to have some leeway. So again, I'm not enough of an enzymologist to give you that specific answer, but I would assume that it's not as rigid as this, that it's like 8 to 9 slice, 8 to 9 slice, but the size is the same. So whether there's movement back and forth in the sequence, that there probably is, but the size is going to be 8 to 9 amino acids. Because if it's 13 amino acids, it's worthless for class 1 MHC loading, because you just can't load it. Okay, does that make sense? So now the peptides have to get loaded into the MHC molecule, and in order to be loaded, the molecule that's utilized to transport it is a TAP protein. And there's two chains, the TAP1 and the TAP2. They bridge the endoplasmic reticulum, and they're playing a role in terms of pumping the peptides into an area adjacent to the MHC molecule. And again, it's amazing in immunology how you go from a simple concept to a complicated way of getting things done. But it's actually, just to walk you through the slide, first thing that happens is you make the MHC Class 1 molecule. Well, this MHC Class 1 molecule needs to bind what protein? Beta-2 microglobulin. So therefore, it basically gets anchored to this protein called calnexin until the beta-2 microglobulin binds to it. Once the beta-2 microglobulin binds to it, the calnexin lets go of it. The Class 1 MHC beta-2 heterodimer now moves next to the TAP protein, and this is kind of like, anyone ever go to the airport to pick up luggage, right? And you have the conveyor belt, right? And there are two kinds of people. There's the people that they just stand anywhere, but there are other kinds of people, they have to stand right where the luggage is coming out, and like they're the first people there, and usually their luggage is last. I mean, but anyway. And they have to be right there, because that way they can get the luggage right away and run and get the first cab and get wherever they have to go. Well, the same thing is happening. Think of the TAP molecule as where the luggage is coming out, because the peptides are being chopped up by the ribosomes into all these different peptides. They're being pumped out by the TAP, and these Class 1 MHC molecules are sitting right by the TAP exit. They're waiting for the peptide to come out. If it's the wrong anchor motif, it's not their luggage. They have to wait for the next one to come. But if they do get the one that has the right anchor motif, now they could leave the endoplasmic reticulum, migrate, and ultimately be expressed on the surface of the cell. Okay, is that clear? Now, the implication is very clear. If you knock out TAP, for example, if you have a mutation in TAP, are you going to be able to express MHC Class 1 molecules? And the answer is no. We'll discuss in immunodeficiency that there's actually an immunodeficiency. They lack TAP, and they don't have Class 1 MHC molecules. And in fact, it turns out they have very few CD8 cells because there's no Class 1 MHC for them to mature off of in the thymic environment. Okay? So this is basically how the loading occurs and the appropriate size peptides. If the MHC molecule has no peptide, can it be expressed on the surface? No, because you don't want an empty cleft to be there that a foreign peptide could lodge into to basically allow the cell to be killed as an innocent bystander. So this is a very, very tightly regulated process in terms of this transport. Okay, any questions? Okay, so now if we go to Class 2 MHC molecule, here the antigen is coming from outside of the cell, and this gets basically taken up into the phagolysosome and digested into peptides. So it's basically going to be digested by proteolytic enzymes that are nowhere near as intelligent as the proteasome. Well, we'll see in a second what the implications of that are. But in parallel, in this kind of coordinated ballet, the Class 2 MHC molecule is being synthesized. It doesn't have a peptide inside. It leaves the endoplasmic reticulum, again, as opposed to Class 1 MHC, which it can't leave until it has a peptide. Class 2, as you said, helper, who cares? You're not as paranoid about what peptide gets into it. It now comes into this phagolysosome, and now it basically gets loaded with peptide, is brought to the surface, and now is expressed on the surface with the peptide that it got loaded with. Now, in contrast to the kind of egg slicer, where you're getting exactly the same size peptides, the proteolytic enzymes in the phagolysosome are not as strict in terms of how big the peptides they are. A lot of them basically need to look for the appropriate amino acids next to each other in order to slice it. So think about this as slicing bread freehand. You basically, thick slices, thin slices, there's a lot of variability, and the end result is that you have a large group of slices that have different thicknesses. And the same way with the peptides that are generated for the class 2 MHC molecule. But does it matter? No, because class 2 MHC is very flexible in terms of peptide size. The same way you don't care how thick the bread is, you know, you just put some marmalade or jelly on it and it tastes fine. As opposed to class 1 MHC in the eggs, where it has to be exactly the same thickness. Now, clearly, loading the peptide is very important, and you do want to make sure that you don't load cytosolic peptides into it. You want to make sure you're only going to load peptides that come from the phagolysosome. And the way that you do that is you basically use a peptide as a placeholder. And the peptide that's used as a placeholder is called the Cleft peptide. And the basic mechanism is that you have this invariant chain. It now folds into the cleft as a placeholder. This invariant chain gets digested, leaving this Cleft peptide that fills this cleft but prevents anything else, any peptide from being put into there as long as it's being transported. And this is, you know, when they ship things, they always stick something in the middle so to protect it, they put a cap or something over it so nothing gets in it. And this is just showing a different figure, again, how it ends up forming the Cleft peptide. And now what happens is it gets placed in the phagolysosome. Here is the Cleft peptide. Here are foreign protein gets digested to peptide. And now HLA-DM, which is an HLA molecule, gets recruited. It helps pop open the class 2 Mhc molecule. Also, the acidic environment also helps. The Cleft peptide gets popped. Now the peptide can get loaded inside of it. And now will be expressed on the surface for T cells to see it. So again, a little bit different approach, but again, because class 2 MHC is going to be helper, you don't have this high level of paranoia. You don't have to have this tight control of how the peptide gets in there and where it comes from. Okay, is there any questions? Yeah.
I always thought that HLA is the same as MHC.
Yeah, the answer is yes, HLA is the same as MHC. But it turns out that, and this is one of the interesting surprises, that in addition to being able to, this particular HLA molecule, its function is not as much to present antigen, but it's to help facilitate this clip from coming out. If you knock out this HLA, DM, you basically have clip that stays in there. It's a lot more, so it's not processed as efficiently. Okay, so now again, class 1, class 2, CD4, CD8. Well, now the question is, what is the T cell receptor seeing? Is it only seeing peptide? Is it seeing some MHC? How much MHC is it seeing? What determines whether the T cell receptor recognizes the antigen or not? And it turns out that this is this important concept called Mhc restriction. And what Mhc restriction means is that in a normal antigen-presenting cell, this is the Mhc molecule, and this is the peptide that's being presented. Here's a T cell receptor. What you see is that the T cell receptor is seeing some Mhc sequences, it's also seeing some peptide sequences, and the summation of this interaction is generating enough affinity to cause recognition to occur. Okay, is that clear? The summation of all these. The T cell receptor just sees residues pointing up at it. It doesn't know whether the residue is coming from peptide or residues coming from the Mhc molecule. It just knows that it has a molecule that binds to that residue, hydrogen bonds, right? So it can't see, it just sees this topology of residues pointing up. It doesn't know where it's coming from. But let's say, for example, here, this T cell receptor only recognizes Mhc, in this case A, for example. If you take exactly the same peptide and you present it in MHCb, true, it could recognize this peptide region, but it doesn't recognize the MHCb region, and therefore it lacks sufficient affinity to bind to that peptide. And therefore, this T cell receptor will only recognize peptide in the context of a specific Mhc molecule. And that concept is called Mhc restriction, which is a critical concept in T cell immunity. So the same exact peptide that my T cells can recognize when my Mhc molecule is presented will not be recognized when your Mhc molecule is presented by my T cells. Okay? And the flip side is, if you take the Mhc alone But instead of having peptide X, you have peptide Y, even though this region of the T cell receptor can recognize this region, but this region doesn't recognize the peptide. And again, you don't have enough affinity. You need a combination of MHC plus the specific peptide to get that level of affinity. And again, you don't get recognition, and this is peptide restriction. You need to recognize the appropriate peptide, obviously, for the T cell receptor to work. Okay, is that clear? Now the question is, well, why is it that foreign, sometimes I could recognize foreign MHC molecules even though they're not ones that I've been pre-programmed to recognize? And this is like when you start getting into some of the hand-waving explanations that immunologists tend to do. But this is basically kind of providing a potential scenario by which this could occur. And we know that because, for example, if I would get transplanted with somebody else's cells, for example, I would reject them. And the question is, well, how can I reject that person's cells if they have a different MHC and my T cells only see my MHC, right? And that's like, clearly one of the major observations. So you can't argue with that observation. So you have to come up with a explanation for it. And the explanation that they generated was, this is a classic situation. T cell receptor recognizes self-MHC plus self-peptide because you get enough affinity from this summation of these interactions to get binding. However, it may be that a non-self MHC can present the self-peptide that I recognize, but it does it in such a way that there's enough interaction between the peptide and the TCR to generate enough affinity that you don't need contributory affinity from the MHC molecule for binding to occur. So it's almost like there's some peptides that are super antigenic, maybe because they have very, very strong charges on them, that they alone could cause T cell recognition independent of MHC. Or it may be that some non-self MHC residues are so polar, for example, that they may generate a large enough binding affinity that even in the absence of a peptide that's being recognized, that alone is enough to tell the T cell you've bound something you recognize and therefore mount an immune response. So this, again, even though the classic situation, you need a summation of peptide plus MHC, it may be that peptide alone may be enough, some peptides, or some non-self MHCs may be enough to get binding, and again, to the T cell to be activated and mount an immune response. And this may be the explanation for why you can mount a very potent immune response against non-self MHC molecules. Okay, any questions? Okay, so now just to summarize, the differences between peptide processing class 1 and class 2, class 1 MHC, endogenous peptide, class 2 is exogenous. Peptide loading in class 1 is an endoplasmic reticulum, the endosome for class 2. Peptide for folding is the antigen-derived peptide for class 1. For class 2, it's Cleft, T cell, CD8, class 1, CD4, and cellular sequelae of presentation, death for class 1, and activation for class 2. Now, one of the really kind of exciting things that have just emerged in immunology in the past few years is the ability to quantify antigen-specific T cells. So, for example, if I wanted to ask the question, how many T cells does someone have in their circulation that recognizes an HIV peptide, how would I answer that question? Up until a few years ago, I wouldn't be able to tell. So, however, this is Altman and Davis came up with this concept that if you basically take synthetic MHC class 1 molecules that are soluble and you basically biotinylate them and then you add strepavidin and now you form a tetramer of these MHC molecules, you could load them with any peptide you want provided this peptide has the appropriate anchor motifs to be bound by that peptide. So now you have a molecule that has MHC plus peptide, 4 of them. You add a fluorescent dye to this, and now you mix them with T cells. If the T cell receptor recognizes that MHC plus peptide, it'll now bind to this tetramer. You need to have at least 3 MHC plus peptides to have enough affinity for the soluble factor to be bound. But now you could, by virtue of the fact that it's expressing a fluorochrome, you could do flow cytometric analysis, and now you could identify what fraction of any population of T cells recognizes any peptide plus Mhc molecule. So you basically could develop a panel of peptides from any pathogen you want in a whole panel of HLA groups, and then ask the question, what fraction, and you could demonstrate beautifully as infection happens, what goes up, what goes down, and really start to get an understanding of what's going on at the antigen-specific T cell responsive level. So this has really served to revolutionize our ability to analyze T cell responses, and especially in HIV, has really allowed to dramatic advances in our understanding of how T cell responds and which ones are important and which ones aren't important or less important. Okay, any questions? Okay, so now this is just going to end with really how things can go wrong. So now clearly, in order for a T cell to get activated, it has to bind only the peptide that it's specific for. Right? That's very straightforward. Now, over 30 years ago, in the United States, there was an incidence of 55 cases. It's called toxic shock syndrome, and I'll show you a picture of what it looks like. And 95% of the cases were in women. 95 had onset during menses. 73% had Staph aureus, and the case fatality rate was 13%, which is huge. Previously healthy women all of a sudden just died from toxic shock syndrome for no reason at all. And these are also young women, which makes it even more devastating. And apparently in other states, it's as high as 25% fatality rate. The question is, what was going on? No one had a clue. And this is what it looked like. I mean, these is basically have toxic shock where the skin basically starts forming these blisters and tremendous level of necrosis, and obviously you have significant infection, very dramatic disease. What was going on? What was the cause? And it turned out that the cause was due to the production of a superantigen by the bacteria, and what the superantigen does is it binds to MHC and it binds to the T cell receptor. In essence, it's like crazy glue, so that instead of only having T cells that are antigen-specific responding, it made all T cells think that they were responding to an infection. All those helper T cells were making all these factors. It was like the cytokine storm, revving up the immune system, causing tremendous amount of autoimmune destruction, and this was the etiology of the disease. It was super antigen mediated, and it turned out that this was due to having a super absorbent tampon called the Rely tampon, which allowed bacteria to grow very efficiently because all this absorbent blood, bacteria said this is a great place to grow. And then since they removed this tampon from the market, this was resolved. But again, it's an example of how an exuberant immune response due to a super antigen that is able to drive the activation and proliferation of T cells independent of antigen could cause devastating effects. Now, in order for antigen presentation to occur, it actually uses what's called an immunological synapse. And what this immunological synapse does is it focuses, these are the T cell receptors and Mhc plus peptide, but now you have a large number of these focused in a very, very focused small area. Why is that important? When we talk about T cell signal transduction, you'll hear about how the interaction of all the signal transduction molecules occurs, and by focusing multiple TCRs in a very, very small area, that allows amplification of signaling to occur. And the way you do this is by having the ICAM molecule and the LFA molecule, adhesion molecule on the outside, forming this kind of three-dimensional, like donut, and the center of the donut are the T cell receptors focused, and the outer ring are the adhesion molecules that now basically push them. It's a lipid bilayer, so they're movable, and push them into focus into this very tight area. And this now allows very much more efficient signal transduction to occur, and that's important in the activation. If you don't have this T-cell synapse formation, you have a markedly less efficient capacity to mount an antigen T-cell response. Okay, but what's very kind of devastating about this is that HIV is always out there trying to figure out how it could take advantage of an immune function to its own nefarious purposes. And what HIV has done is it actually has co-opted this process. So here you have ICAM, LFA, TC. Oh, I flipped this around. Okay, so I flipped the top and not the bottom, so I messed this up a little bit. I apologize. So basically, this is the normal T-cell receptor synapse the way it's supposed to be. What happens with HIV is you have the target cell, the effector cell, but instead of focusing the T-cell receptor, you now focus HIV. And what you see here on this kind of magnification, here you have your CD4 molecule here. You have, okay, cool. No, I take it back. I didn't, oh, okay, this is what happened. This end got messed up. Okay, bottom line is you have Mhc Class 1, CD3, normal immunological synapse. Here, you have ICAM1, but now what it's focusing is the HIV. So GP120 and GAG are focused over here against CD4 and CXCR4 or CCR5, and this now allows HIV to zip across and infect cells through cell-to-cell interaction. So again, it's taking this kind of tight membrane interaction that's utilized for signal transduction, and is used it as a way of allowing HIV to get transmitted from cell to cell. Okay, so basically in this lecture, the question we started out with was what are the structural bases by which Mhc molecules present peptides to the T-cell receptor? And I think that we talked about the alpha chain in beta-2 myoglobulin class 1, alpha and beta chain for class 2, and again, the implications in terms of flexibility of the heterodimer of the alpha class 1 and the alpha beta of class 2 determines what size peptides and how tightly the peptides are bound. We discussed how endogenous peptides are targeted to class 1, proteasome, endoplasmic reticulum, class 1 binding, and then only then can it migrate to the periphery, and MHC class 2 binds CLIP, and the peptide transfer occurs inside the vacuole. How does T-cell receptor see the peptide and MHC? It sees a combination of both, but there are exceptions where some peptides can be very high affinity, some MHC can be very high affinity, and that may account for ability to generate a graft rejection. And the structural basis of CD4 T-cell MHC class 2, again, as I mentioned before, CD4 binds to the piece of the class 2 Mhc molecule, and CD8 binds to the alpha 3 region of the Mhc class 1 molecule. So again, thank you very much for your attention. I hope you know a lot more immunology now than you do this morning. Thank you very much, and see you tomorrow.